So our next speaker is Dr. Jenelle Dunkelberger from Topigs, Norsvin, where she's a geneticist. So Jenelle holds a bachelor's degree from Northwest College in Orange City, Iowa, and earned a PhD in genetics from Iowa State University. She's active in publishing papers and giving talks and proceedings at conferences on genetics, animal genetics related topics, and is particularly interested in the role of host genetics on the response of disease challenges. So she's active in research, obviously, and is pursuing the area of research in her current position as leader of the Global Health and Behavior Research Platform with Topigs Norsvin. And apparently Jenelle lives not too far from here with her husband and her son. So I'll pass it over to you, Jenelle, and interested in hearing your talk. There we go.
Hello, hello? Yeah, that sounds a little bit better. I have a pretty loud voice, so maybe you heard my little spiel there anyway. Yeah, good afternoon, everybody. Not sure where to clip this. Yeah, today I'm going to talk to you a little bit about some of the work that we're doing to breed robust sows for commercial conditions. And I think we're all sitting in this room right now because we're interested but also concerned about sow robustness within our industry. We've talked a lot about that already, including different reasons for sow mortality, but we're shocked and concerned about this increasing trend that we've seen, particularly since about 2014. One of the things that we haven't yet discussed, though, is that there is this suspicion, right, that sow mortality at the rate we're seeing currently is kind of a US-specific issue. I'm not sure that I'm necessarily convinced of that, given that we do see increasing sow death loss in other countries too. But when you look to the level of the sow death loss between the US and comparable markets, that difference is quite striking. I think a really good example of this is if you look at our current industry average for sow mortality for the US versus Brazil, right, which is a great comparable market, large-scale production, similar production practices, similar nutrition, yet their mortality rate is approximately half that of what we're seeing in this country. And actually, Dr. Yeski had an even more updated number than what I have, showing that it's now currently at around 15%. So when you look at these figures, you think, okay, we've got similar genetic products in both locations. So clearly there's got to be something else environmental that's contributing to this. So what are some of those things? Mortality is obviously devastating regardless of the stage of production, right? But at the sow farm level, this is particularly problematic because sows are extremely expensive. And there's a lot of labor involved in figuring out what to do when a sow dies. You've got gaps in production. It's challenging to get that sow out of the farm. And all at the same time, we're already struggling with this labor issue within the country. It also certainly has a negative impact on sow farm financials, right? Particularly those animals that are falling out prior to P3 and for which the producer has not yet made a return on that investment. It's also inconsistent with sustainability goals. Actually, I'm a little bit surprised this hasn't come up a bit more in presentations today. But I mean, we can't deny this either. We're getting a lot of pressure as an industry to start meeting some of these goals to become more sustainable. Mortality is an animal welfare issue when that animal is dying for involuntary reasons, right? And of course, the whole theme of sustainability is putting pressure on us to produce more with less. Mortality is absolutely counter to that. So we need to have robust sows, and we define that as sows that are going to thrive not just in their current, but also their future production environments, leading to more sustainable pork production. There's been a lot of discussion on this point already, talking about this disparity between what's happening at the nucleus level and what type of conditions are we selecting our most genetically elite animals in compared to what's happening at the commercial level. That's absolutely true, right? We keep our most genetically elite animals in nucleus herds where we do everything that we can to try and optimize the environment for those animals. But we do have really good reasons for doing that, right? We have to maintain a high health status because those animals are being moved around, and we need to be able to properly evaluate the full genetic potential of these individuals. But a consequence of that is we don't get longevity phenotypes. We also have a very high turnover rate of those animals, and that's because we need to be constantly evaluating our most recent genetics. That means that these animals, we just don't give them a chance to express their longevity phenotype properly. We also don't have the ability to collect robustness phenotypes, right? In the absence of health challenges, they get the best management, they get the best feed. And as we've talked about several times so far, we just can't improve what we don't measure. So how do we get around this gap between the nucleus level and the commercial level? At the nucleus farm, we routinely collect tissue samples from every animal that we use for genotyping. But we don't have the opportunity for robustness data, as I just mentioned. At the commercial farm, oops, I think that was the wrong button. We do have opportunities to collect robustness data. So if we collect tissue which we can genotype, we can actually link these herds via genetic relationships. When we do that, we actually have the ability to estimate breeding values for traits like longevity and robustness for both of these populations. So this is the workaround that we use in order to calculate breeding values for traits like longevity. And I got a point in Gustavo's presentation earlier. One of his summary messages was we should focus less on longevity. Oh crap, I have this slide which I'm going to make a point about longevity. But I think, to your point, was we shouldn't focus only on animals that are dying, but animals that are being culled. It's the whole package. Sow retention should be the main goal. And I agree with that. And that's consistent with our approach, because actually the breeding values that we're estimating, call it longevity here, but what it really is stayability. Is that animal retained until parity 2, yes or no? Or is she retained to parity 5, yes or no? So we estimate breeding values for both of those traits. They're heritable and part of the index. And this is a 5-year genetic trend showing progress in that breeding value for longevity to parity 5. You can see that's moving in the right direction. We've also talked about different reasons for sow mortality. And I'm sure you've seen this figure a number of times, but this is one adapted from the Pelvic Organ Prolapse Project report coming out of Iowa State by Jason Ross. But I think what's really striking about this is that when you look to the different reasons for sow mortality, actually the top 3 reasons explain over 80% of why sows are dying. So it's unknown or other, Leman downer, and vaginal and uterine prolapse. We've heard those terms come up a lot, sometimes in a different order, but obviously relevant. So I talked about how we have breeding values for longevity as part of our index to address that number 1 category. Torin went into a lot of detail describing our approach about how we are tackling that number 2 issue of feet and leg structure. So I'm going to spend the rest of the time talking about how we're addressing pelvic organ prolapse. So I'm going to refer to vaginal or uterine prolapse simply as POP for the rest of the presentation here. You know what that is. You know it's devastating, particularly because it always means that you're going to have to make a decision typically between euthanizing that sow if it hasn't already died naturally. But it's also considered typically that this is something influenced by multiple different factors. You know what those are too. This is definitely not an exhaustive list, but we see things like mycotoxin, vitamin deficiency, water quality issues, sleeving and induction practices, disease, and genetic background is one of the things that has been suspected as well. So that's something that we wanted to dig into further for the first study that I'm going to share with you. where the objective was to estimate the heritability of POP in a purebred Topigs Norsvin maternal line. So the data used for this study was collected on purebred Topigs Norsvin females from a single commercial multiplication farm located in the US. And the response variable that we analyzed was removal due to POP versus removal due to something else. We had a lot of available data. So in the full data set, we had records on over 16,000 sows. And in what I'm calling here the reduced data set, we had matched case control pairs on 986 animals. And the way that we defined the controls to match to the cases in this data set was by selecting females of the same farm, the same parity at removal, and inseminated during the same month and year as the case observation. The nice thing about a case control analysis is because you've already pre-corrected for the fixed effects in the sampling scheme, you don't need to do that again when you fit the model. But we did, of course, have to account for those things when using the full data set. We wanted to run a genetic analysis, and so we used 3 generations of pedigree in order to account for the relatedness between individuals. And this is what we get. So we looked at analyzing prolapse in 4 different ways, using the full versus reduced case control data set, and analyzing prolapse as either a binary or a continuous variable. To summarize these results, we saw that actually the standard error remained consistent across all 4 scenarios. And actually using that case control data set, which consisted of only 12% of the data of the full data set, did not decrease accuracy of heritability estimation. We saw that the data set structure, so whether we used the full versus case control data, had a greater impact on heritability estimation when we analyzed as a continuous versus binary variable. So given the effect that those estimates remained more stable or consistent by analyzing as a binary trait, and it's always nice to use full data whenever possible, our best estimate of heritability from this analysis is 22%. The second objective of this study was to look at actually validating breeding values for prolapse in an independent and related population. So we see that prolapse is a heritable trait. So can we show that that works in practice by seeing the effect of breeding values in an independent population? The training data set is the same population that I just described. And the validation population consisted of females that were the same breed and also a same genetic line from Topigs Norsvin, but from a different farm in a different geographic location. This was actually a nucleus farm. And the question that we wanted to ask here is, are breeding values for POP of a sire predictive of the incidence of POP in his offspring? So we selected sires that had 10 or more daughters within the validation population and then estimated breeding values for all of those sires. This is kind of an overview of how that works. So within the training data set, all of those animals have both genotypes as well as phenotypes, which allows us to estimate what we call the SNP effects for each of the different genetic markers that those animals are genotyped for. The animals within the validation just have genotype information, but we can use that by looking at the genotype that an individual carries at each genetic marker, plug in the SNP effects derived from the training population to calculate what we call a genomic estimated breeding value. So this is what the results look like. We've got the sire breeding value on the x-axis and then corrected offspring performance on the y-axis, where that indicates % removal rate of his progeny. And what we see is that there is a nice positive trend here indicating that the higher the sire breeding value, the higher the incidence of removal among his progeny. So these breeding values are predictive of incidence of prolapse in the progeny. So to summarize this part of the study, we saw that an increase in sire EBV was associated with increased incidence of POP among his daughters. We also saw that the 95% confidence interval for the slope that was estimated contains the value that we would expect of a parent-offspring relationship, which is good validation. So therefore, the sire EBV estimated using individuals from a purebred training population was indeed predictive of POP among his progeny in an independent population. So breeding values work. The conclusion from the first part of this study is that POP in sows is moderately heritable. This was actually the first study to show that, and therefore can be addressed via genetic selection. As a result of this work, we made the decision to start estimating breeding values for POP on an ongoing basis and actually added that as a new trait to our selection index. So that decision was made in 2021. You can see that in terms of genetic trend, it was already moving in the right direction prior to that, but this steep increase or decrease that you see here indicates that with the decision to implement explicit selection based for uterine prolapse, that we will expect the incidence of uterine prolapse to decrease at an even faster rate. So some remaining questions we had from this work is, would the heritability estimate of POP change if we reanalyze these data actually using genomic rather than pedigree information? And then secondly, are there specific genes or genomic regions that are associated with susceptibility to POP? So to do this, we had a collaboration with Iowa State University. I had an intern last summer by the name of Vishesh Bhatia, who is a PhD candidate at Iowa State studying under Dr. Jack Deckers. He spent some time in the Netherlands, took the same data set that I just described to you, and used the genomic information to run analyses with those data. And he was partially sponsored by the Iowa Pork Industry Center Pig Survivability Grant and advised there by Jason Ross. So his first objective here was to re-estimate genetic parameters using the genotype data. So altogether, Vishesh had still over 14,000 records that he used for his analysis. He limited the data set to include only sows between P2 and P6. Again, these were purebred Topigs Norsvin females, and they all had genotype information available from either a 25K or 50K SNP panel. So as a reminder, this was the best estimate of heritability that we derived based on using pedigree information. And then when Vishesh reran the analysis, he estimated heritability at 35%. So first of all, this is why we genotype, right? It's a testament to the power of genomics. We're just that much more able accurately to capture relatedness between individuals. So the conclusion from this is that POP appears to be even more heritable than what we had previously realized. The second objective of his work was to identify genes and genomic regions associated with susceptibility to POP. So we've seen that it's a heritable trait, meaning that it has a genetic basis. Can we identify any potential candidate genes underlying that genetic variation? So he did this part of the study by conducting a genome-wide association study, where I'll show a series of these GWAS plots here that indicate statistical significance of different regions of the genome. You see the chromosome is listed on the x-axis, and then the height of the different dots here are representative of the percent of total genetic variance that those regions explain. Anything above this red dotted line here is considered significant, and each dot actually represents a 1 megabase window, which is actually a group of different genetic markers. Altogether, Vishesh identified 6 significant regions, which when summed add to just over 9% of genetic variance. So although we do see specific regions popping up here, Individually, they explain a relatively low proportion of total genetic variance, still leading us to conclude that this is a trait controlled by a number of different regions with smaller effects on the trait. Nonetheless, let's look to some of these regions and see what kind of interesting genes they contain. On chromosome 1, he did identify a candidate gene there, ESR1, which you'll recognize as the estrogen receptor. This makes a lot of sense, right? Because it's got roles in pelvic floor support, smooth muscle, and it's actually also been associated with prolapse in both humans and pigs in previous work. On chromosome 3, he identified a number of candidate genes associated with calcium level. That also makes sense because there were previous associations of calcium level with uterine prolapse in other species. On chromosome 7, he identifies a gene which binds to this protein called fibulin 5, plays a role in pelvic organ support. On 10, he sees TGF-beta 2. You can see here, roles in uterine development, and then these terms of smooth muscle appear again. On chromosome 12, he finds genes associated with cellular structure, and again, very interesting to see that actually this family of genes has relationship to POP susceptibility in mice, actually. And lastly, another gene here with a role in calcium and phosphorus homeostasis, again making references to calcium. So to conclude this part, results from his study confirm that there is a genetic basis to POP. Results from the initial study using pedigree data concluded that that heritability estimate is 22%, but when you apply genomic information, that heritability estimate increased to 35%. And the use of genomic data is really useful and important because it increased the estimate of heritability, but it also reduced the standard error. And in terms of looking at the genetic basis of this trait, in general, we see that this is a trait controlled by many regions with small effects, although some interesting candidate genes were identified. And you can see all of the details of this work if you're interested in digging into a little bit more in this publication that Vishesh published in Frontiers in Genetics that came out this spring. So some remaining questions. Everything that I've shared with you so far is research that we conducted on the purebred female. But of course, what's more relevant for the industry is looking at the impact in the F1 female, given that that's the majority of females that are represented within the industry. So that's the next question. What's the heritability of POP for commercial F1 females? We also see from some previous work from the Pelvic Organ Prolapse Project that there are a number of non-genetic factors that have been associated with susceptibility to POP. So of the different things that were evaluated in that project, there were 4 factors that were significantly associated with susceptibility to prolapse: water treatment, bump feeding strategy, perineal score, and body condition. So our next question was, can we validate the effect of one or more of these factors in an independent F1 population? So we had another intern to help address some of these questions. We had a student from Iowa State, Tricia Veldhuisen, who worked with myself this summer. She's currently finishing up her final year of her bachelor's at Iowa State. And Tricia spent a lot of time on farm this summer, going between 2 huge systems in the Midwest, collecting data on a high volume of animals. First, to estimate heritability of POP. So to address that, she collected phenotypes in the form of all instances of prolapse, so regardless of the parity or stage of gestation. She also collected tissue samples from all cases and controls. In order to estimate the effect of some of these non-genetic factors, she measured body condition score, caliper score, and perineal score for every animal as it entered the farrowing unit. Dr. Yeski already showed this figure. It's the same here. She used this 5-point body condition scoring guide where we consider 3 to be the ideal score. And she used the caliper, which we've already discussed today, a tool that you place on the back of the animal to look at measuring the angle. She also recorded perineal score according to the scoring system designed by Zoe Kiefer at Iowa State, where each animal was assigned a risk score for prolapse, essentially, a score from 1 to 3, 1 which is indicative of little to no risk of prolapse, 2 is moderate risk, and 3 is high risk. And this is what we see in terms of the incidence of prolapse within these systems. So again, she collected data at 2 farms and had a large number of total observations. But in general, the incidence rate of prolapse was low. So we only saw 33 animals that prolapsed versus over 3,800 animals that did not prolapse for an incidence rate of just less than 1%. But if we look at the relationship between the number of prolapses by perineal score, I think the results here are quite interesting, where we see that the most prolapses happened for these animals that had a perineal score of 3, right? And it's actually quite linear here in terms of the relationship between these variables, where the sow that had a worse perineal score in late gestation was more likely to prolapse. And that exactly agrees with the findings by Ross in his report. When we look to the breakdown of the perineal scores, again, we had 2 farms, so A and B here. We can see that the majority of animals had little to no risk, but we do see some differences between farms A and B, which I find really interesting. So the percent was twice as high for a moderate risk score for the animals in farm B relative to farm A. And in general, low incidence of animals with these high risk scores. So as a result of this category, I actually combined these 2, perineal score 2 and 3, to analyze perineal score as a binary trait, looking at a favorable or ideal perineal score versus a non-ideal score. And so this is what we see when we estimate the effect of body condition score on perineal score. And again, we see the trend here is really quite linear, right? So the animals that had the highest incidence of non-ideal score were those thin animals. And these animals that were fat had the lowest incidence of non-ideal score. We see a similar result when we look to the effect of caliper class in perineal score. And again, this is highly significant effect. Tells the same story, right? Those sows that are considered thin have the highest incidence of a non-ideal score. So to conclude, these results, these validate previous findings from that pelvic organ prolapse project. We see that those sows that had a higher perineal score or lower body condition during late gestation were also the animals that were most likely to have POP. So next steps for this project, first to submit samples for genotyping. This is all very fresh because she just completed the project. We want to do that, again, with the objective of estimating the heritability of perineal score and POP in a crossbred population. We also want to estimate genetic correlations between body condition score, caliper score, perineal score, and POP. And then finally, to look at assessing the predictive ability of these breeding values that we develop based on a purebred population to predict the incidence rate in a crossbred herd. So overall conclusions, sow longevity and susceptibility to POP are heritable traits. And I've shown heritability estimates for both of these traits. We see that there is genetic variation and they can be improved via genetic selection. Because of that, we can and we should do that and address it through the breeding program. That's our responsibility as a genetic supplier, and we've done that. But if we look to trying to understand the genetic basis of POP, we see that it's very complex. Not just complex as in complicated, but it's caused by many regions with small effects on the trait of interest. So going for selection based on genotype at a single gene associated with prolapse is not going to work. You really have to estimate breeding values using the SNP effects across the genome. And that's how we're addressing this number 3 category. Again, I talked about how we use breeding values to address longevity or stayability to parity 2 and 5. Shoulder sores and body condition at weaning are also ways that we address this unknown and other category. And Torin described in very good detail how we use actually CT images to score osteochondrosis and all of the other 32 traits that we score in order to address this number 2 category. And although we see that longevity and POP are heritable, please keep in mind that the majority of phenotypic variation is still caused by the environment. That could explain differences that we see in sow mortality rate and POP by country and geographic region, as I alluded to in the beginning. But it also confirms that we can't stop looking for these non-genetic solutions to approach this issue. We have to keep our foot on the gas in trying to identify and implement those non-genetic solutions as a way of supplementing this approach. And managing body condition, as I just described through that last study, that can be one approach or way of addressing that. So with that, I'd like to say thanks to all of the researchers that you see here that contributed to this work, and thank you very much for your attention.
Thanks very much, Jenelle. Very interesting. A good overview of the work that you're doing. I'm just going to ask a quick question right away, and we'll see if there's some other questions in the audience. But I guess one of the things I was interested in is the fact that you're using relatively low-density SNP panels, so 25K and 50K. Is there any plans to use a higher density SNP panel to see if you can explain a bit more of that variation using specific markers?
I love that you asked a genetics-y question. Thank you, Mike. Actually, we used to use a 50K SNP chip routinely and have since gone down. Yeah. So we used a 50K in the past, and now we use a 25K routinely. So we cut that in half. And we do that for practical reasons. It's less expensive, but we're able to do that without sacrificing the prediction accuracy. So it's good justification for doing that. But the nice thing about this, the genetics approaches too, is that we have the ability to impute up. Right? So even though we genotype across 25,000 genetic markers, we can impute up to 660K or even to sequence.
Yeah, that's what I was going to ask. But have you done some validation genotyping to determine whether your imputation's working?
Yeah, and we have people in here that are the experts on that.
Okay.
Yes.
Okay.
Yep. Any other questions?
Thanks, Jenelle. So my question is, the work that Vinish did, many identifying those genes, 6 of them that you went through, many of those had coded for proteins that were important in prolapse in other species. Did he ever go back to that work on those other species and see if that same 6 family of genes popped up as important as well?
Like all 6 together?
Well, I don't know if there were others that were identified in his population that were then also identified in the mouse model, for example. You said that there was one gene that was particularly noted. Just wondering if any of those others that were in the mouse model rose above that line too.
Yeah, he didn't take it from that direction. Yeah, so he first started by looking at what regions are significantly associated in this data set, and then went to see if he could find biological support for those regions from the literature and other species, but didn't go the opposite direction, no. And I think that's, it might be interesting to have a look at, but at the end of the day, it's the significant regions that, we're gonna be most interested in.
Yeah, and I appreciate too the fact that you talked about not the genetic component, but also the component? Because I think early on, we were thinking there might not be a genetic component, because you could have the same genetics on two side-by-side sow farms fed out of the same feed mill, and one was running 14% prolapse and the other one was running 4%, right? So how do you describe that? So I think early on we were thinking that there was not a genetic component to it. So I'm glad that you were able to show the heritability and demonstrate that, and to begin to select against that. So I think that's going to be important. Thank you.
Thank you, Chris.
Any other questions, comments? I just wanted to maybe just comment on the fact that you raised the point of sustainability, and I agree completely that I think that we may need to be starting looking at the impact that some of these inefficiencies have on sustainability, whether it's, maybe we're, as an industry, flying a little bit below the radar because we have people in the cattle industry who are pumping out greenhouse gases and that sort of thing at a relatively high rate compared to us. But at some point, I agree that we need to be starting to look at, okay, when we have inefficiencies, when we have losses, mortalities, and all of the resources that went into that animal before the mortality, we need to be considering what is the cost of that at a resource level, whether it's greenhouse gases, whether it's emissions, those sorts of things as well. So I'm glad that you raised that, and that might be something that we need to highlight in another workshop at some point. So
There you go.
Relating to this, internally we identified this issue as a non-competitive issue because it chases people away from the farms. It needs to be solved, and not just by us, but by others. So that's why we try to be open, and we're open for collaboration if we want to go the further fine mapping or whatever. So if there are competitors who are interested, then we can work through Jacques Dekkers or the directly together. And that is, I think, sustainability. That is creating an image in the industry which we need to have.
yeah, go ahead.
Just a question about the environmental component. The demand for culling, and I enjoy that. I appreciate that you include culling with mortality because the two are related. But the demand for culling based on breeding group size and supply of gilts and the like, is that included in the environmental pressures, or is that another category?
Yeah, so what I showed in terms of estimating heritability for longevity or stayability, however you want to define it, that's just looking at the genetic component. Yep.
All right, we're a little bit into our break, so I'll entertain one more question and then we'll maybe shut it down.
Thanks. Did you look at perineal score versus genotype? You looked at genotype versus pelvic organ prolapse, but just seems like that perineal score is going to represent more animals. So just your thoughts on that.
You're exactly right, and that's one of the motivations for us adding the perineal scoring to the protocol, is because although prolapse is a really frustrating trait, at the end of the day, it's still low incidence when you go to analyze it statistically. So indeed, by adding the perineal scores to the protocol, we do get more observations in order to really facilitate the analysis. For that last part of the study, we have all of the tissue collected for genotyping, but we still have to submit them for genotyping. So I don't have results on that yet, but hopefully stay tuned that we should have that soon.
Excellent. So thanks very much, everyone. Let's thank Jenelle once again for an excellent presentation.